My portion of today's talk is titled From Clinic to Discovery, Maximizing Standard of Care Biospecimens. And to lay the foundation for that, we're going to back to basics. Where do patient biospecimens come from? And of course, the answer is patient material comes from patients. So this flowchart represents the standard of care clinical workflow that patient tissue goes through. So it starts with the patient procedure, the surgery or biopsy where tissue is removed. That then is dropped into formalin within minutes of removal from the body to create an FFPE sample. From that, the H&E slide is cut and stained. Any diagnostic tests, if they've been ordered, are performed, and the information is written up in a pathology report. Then those FFP are stored. In the US, the number is 10 years. The number of the years can vary by country, but this concept of FFP are stored in case of occurrence so that they can be pulled and re-examined is a global standard. So if this is what happens without research interests represented, when we come in to represent like, okay, this tissue, in addition to its clinical value, has research value, there's really two windows of opportunity to collect tissue for research purposes. The first is right at the very beginning, that few minutes long window after tissue is removed before it's dropped into formalin. If there is excess, if there's enough to share with research, we can come in and collect then. And the second window of opportunity is at the end of the workflow. Once that 10-year requirement has been met, samples can be released for research purposes at that time. So if those are the two windows where the samples come from, the data comes from these two steps. The first is any diagnostic tests. And unfortunately, the reality here is that personalized medicine has not made ingrounds into clinical adoption as far as we would all like, right? And this is kind of as an industry, what we're fighting for and moving towards is further adoption of personalized care. The Personalized Medicine Coalition did an influential paper in 2022 focused on advanced non-small lung cancer patients and found that only 52% of those patients actually got the diagnostic tests that would inform a personalized treatment decision. And they chose advanced non-small lung cancer because that's one of the areas where we have made the most advancements, right, into clinical adoption of personalized medicine strategies. So from a research need perspective, if this is where our biomarker data is coming from, at best, we're seeing it 52% of the time. And that's kind of your best case scenario. So it ranges from 0 up to 50. And then, of course, that 10-year-old archival retrospective acquisition now sets the clock back 10 years ago. So that's one data gap, and we'll talk about more gaps on the next slide. And then, of course, there's also the pathology report, which is your source of truth for diagnosis, for stage TNM, for cellular subtype. So listing out the data needs of researchers on the left, some of those data needs can be filled by that data from the standard of care, the path report, the medical record, if there were diagnostic tests. But there's a gap. Researchers need to understand the cellular content of each individual sample and the specifications of each individual sample. And that's a need that isn't shared with clinicians focused on patient care. So we need to come in as a biobank with strategies to fill that gap. There's also kind of a secondary gap here, which you notice if you start paying attention to the timeline, right? That first sample collection opportunity, I said it is minutes long when tissue is removed from the patient before it's dropped into formalin. That's your window of opportunity to identify that it's good for research use and divert it onto a different workflow to make different sample matrices. But when at the point you're making that decision and rediverting tissue onto a different workflow, the path report hasn't been written yet. So the amount of information you have at the time of that collection is actually fairly low. And that's a place where biobanks then, because you are buying prepared, preserved, banked samples, there's now been time for the path report, the diagnostic test results, right? All of that data to be generated and then brought into and matched up with the applicable samples. And our strategies to fill this data gap need to be tailored to each sample matrix. So we're going to talk through the most common tissue sample matrices individually. The first step is formalin fixed paraffin embedded. This is FFPE. Our strategy here for biomarker data is sequencing IHC and PCR to get PD-L1, MSI, HPV, and NGS data. We get tumor percent and necrosis percent to understand the cellular content. And we provide dimensions, tissue XY, tumor XY, an estimated number of sections remaining to get at that Z depth. And we're very proud of our partnership with PathAI to allow us to get further level of precision and quantification on our tumor percentages. The picture on the left represents the workflow that we've been using for well over a decade, which is a triple pathology review. And this generally, for this particular sample, we get ranges like 5 to 15% tumor, whereas our new AI quantification tools allow us to get very, very precise. So this exact H&E has 8.65% tumor. Next sample type is our viable cells. So these are dissociated tumor cells, dissociated normal tissue cells, and bone marrow mononuclear cells. Here, right, our IHC strategy that we use on our FFP is no longer applicable, and we pivot to flow cytometry in addition to sequencing and PCR to address that need for biomarker data that diagnostic tests do not fill. And then to understand the cellular content, we're again leaning on flow cytometry. We have a standard panel. So your EpCAM positive percentage gets you your tumor cell fraction, your CD45 positive percentage gets your immune cell fraction, and then the panel contains further markers then to further understand the immune cell subsets. Sample specifications for viable cells are your post-thaw cell counts. We report that out as a min and a max. The difference between those two numbers is really whether or not you wash the cells and how many times. Lots of washing, you'll see closer to the min. Less washing, you'll see closer to the max. And then a post-thaw cell viability. And again, thinking through this, where this fits into the standard of care workflow, these viably cryopreserved cells really actually only come from that first collection opportunity, right? The minutes after the tissue is removed from the patient when it's still fresh, before the site drops their portion of the material into formalin, we can divert some of it onto a different workflow to make these viably cryopreserved dissociated single cell suspensions. And the reason that this cellular content matters so much is there's incredible variability from one patient to the next. The chart that we're looking at on the left, every dot represents a unique tumor that we have collected, dissociated, and run flow cytometry on. Our y-axis is our tumor cell percentage, so the upper bound being 100%, lower limit being 0%, and the clusters along the x-axis represent different indications. So you can see, while there are some patterns that certain indications tend to, cluster at a particular point in the spectrum, any individual tumor, any individual dot can be as high as almost 100 and as low as almost 0. So that's why having these sample-specific cellular content information is so critical. Because every tumor is different and every patient is different. And if you're studying, say, TILs in the tumor environment, you need a sample where TILs are present. And the only way to know that is to, have that value for that particular tumor. And we feel really confident that these patterns we're seeing are true across the board, just because the sheer scale of collections, associations, and flow cytometry we perform. We first launched our dissociated tumor cell product in 2010, and that we did 100 that first year. The subsequent years, we were doing between 200 and 600 collections and dissociations a year. And then 2018 up through the present, we're doing over 1,000 to over 3,000 viable cell collections and preparations a year. And to further expand kind of our viable cell strategy, just this calendar year, we've launched a new tumoroid product in conjunction with our partners at Thermo Fisher and their OncoPro Media system. Tumoroids are a long-term, viable 3D primary patient cell model system. So that image on the left is IncuSite live cell imaging of tumoroids growing in culture. And to address the biomarker data need for tumoroids, we're able to kind of piggyback off of our quality control data, which is we run whole exome sequencing and RNA-seq on the original patient tissue and the finished tumoroid cell line in order to compare and confirm that this tumoroid still represents the same genetic diversity seen in the original tumor. Which then means you have the whole exome sequence available to see your biomarker data to select which lines you'd like to receive. The cellular content of tumoroids are only tumor cells. They do represent the full heterogeneity of tumor cells, unlike cell lines, which can be kind of a clonal population. If 15% of the original tumor had, say, a KRAS variant, 15% of the tumoroid cells also have a KRAS variant. And then we have paired samples available for co-culture to address the research need for other cell types, such as immune cells or fibroblasts. Sample specifications are in the C of A, viability, passaging, cell count, and line-specific notes from our lab team. And this product is intended to help address the translational gap. So here we're looking at drug dosing data from 2 different tumoroid lines, and you can see sometimes they respond similarly, sometimes we get a differential response. And this is really kind of the primary use case for the tumoroid modeling system. The last tissue sample type we're going to talk about is flash frozen. And here the kind of crux of the strategy is collecting flash frozen paired to FFPE so that we can put the frozen in the freezer, leave it there, and then get all of our cellular content data from our analysis of the QC of the FFPE block. And for the sake of time, those are all the sample types we're going to touch on today. But we collect kind of the full gamut of what's available. Certainly blood is a rich source of data. And we also strive to collect matched samples whenever possible. So if we're making a tissue collection, we always pair it with a blood collection so that we're able to just further expand the types of scientific questions that our biobank is able to answer. And the impact to all of this is to accelerate science. We're really, really proud of our 1.58 NCE to order ratio. And all of the products we've discussed are, banked inventory samples that you could expect to receive within days of receiving a PO. And finally, I'm going to end today with kind of an announcement of a new program that we've started. This is the Discovery Legacy Program. It is intended to help broaden the availability of this biobank to more researchers. There are over 200,000 legacy samples that are part of the program. 30 to 40% of those samples have patient follow-up data. So because some time has passed, we're able to go back, revisit the patient's medical record and see what their response to treatment was, which again, right, just expands the type of research that these samples are able to help answer. So we talked about patient material starts, right, by coming from a patient and their donation. And that kicks off kind of this flywheel of innovation. Next up is biobanks come in, fill those data gaps by characterizing the samples, which then empowers scientific discovery, ultimately leading back to patients with improved therapy and improved diagnostics. And if you have any questions, you can reach out to us at info@dls.com.
Maybe we'll start with you, Meredith. A question here. Isn't the tumor percent redundant to the more detailed pathology report? And maybe an addendum to that with another question here. Are there other things that AI can quote unquote see in an H&E image, for example?
Yeah, sounds good. Well, we'll start with the easy one, which is the first half of the question. Pathology reports are very detailed, but that detail does not meet that research need of tumor percent of each individual sample. The detail that you can expect is a very kind of paragraph format, qualitative description of the tissue that's received by the lab in formalin, and then another qualitative paragraph format description written by the pathologist of what they see when they look at that tissue under the microscope. What that does not include is if there's, say, 26 samples generated from that one procedure, they don't list the tumor percent of each of the 26 samples because it doesn't have relevance to patient care. AI, though, opens up a whole new world of possibilities. I mentioned how we are using AI to define tumor percents more quantitatively, but that's really just the tip of the iceberg. There's kind of five main things that I'm excited seeing AI do. The first is defining regions. So like tumor region, that's our tumor percent. Cancer stroma being another region. Necrosis, right? Those are all, think like millimeters squared. What area of the cell or of the whole slide image is represented by those regions. Second thing AI can do, AI can count cells and classify them. So, like how many lymphocytes are in this block, how many tumor cells are there. And then once you start pairing those two together, things can get really powerful, right? Like how many lymphocytes are within the tumor area and other questions like that. AI can score stains. So think like, those basic diagnostic test biomarkers can be scored via HI, which again is a way to move towards even more standardized, even more quantitative data for our patients. But the two that are kind of most interesting and most like futuristic feeling, AI can predict unattested biomarkers. So, for example, a slide that has never been tested for MSI, but this H&E image is a very routine stain that's done, internationally already. We would be able to predict this biomarker status based only on the H&E, and then have a much more informed decision about which patients should be reflexed to further diagnostic testing, which could help control costs and ultimately drive deeper clinical adoption of personalized testing, in addition to kind of all the research potential of identifying biomarkers, which brings me to the last one. AI can identify novel predictive biomarkers, patterns in the cells and what they look like on the slide when paired with treatment and response data. The AI algorithms are capable of identifying things that help predict response that we wouldn't have picked up on using any other techniques.
Meredith, for this one, you mentioned the tumoroids. Can you comment on how those would be different from a just a cell line, for example?
Yeah, that's a great question. So cell lines have been around for several decades. If anybody hasn't read The Immortal Life of Henrietta Lacks, highly recommend as a book reading. Covers the history of one of the first cell lines. But what cell lines ultimately are supposed to be is they're supposed to all be clonal identical children of a single parent cell. So not the full tumor, just this one cell. And of course, the reality, as cell lines have been around for decades, is that any heterogeneity present has been introduced by genetic drift. So it's actually further removing the cell line from representing that original patient tumor. So while cell lines are a powerful tool in our research toolkit, and we still continue to use them in our services work internally, they have failed to fill the translational gap, which is 90% of clinical trials end up failing, despite the fact that 100% of clinical trials had great lab data to justify starting the clinical trial in the first place. And that's where tumoroids are different. Tumoroids don't represent a single tumor cell, they represent the full tumor, in hopes that they will be able to be a more predictive lab study of future clinical response.
Okay, fantastic. A question here too, along the same lines. Are those a service only offering then, the tumoroids?
No, both inventory and service. So if you want to work with tumoroids in your lab, we'll ship them to you. If you would like to pay us to work with tumoroids in our lab, we're happy to do your project design as well.
Someone has asked, do you feel that cryopreservation can be improved with better samples downstream from any new technologies in this space? And I know we've talked a bit about new technologies and advancements, and maybe any other general commentary on things you're excited about as we move forward. Meredith, you look like you're ready
Yeah.
to
Yeah,
tackle
I
that one
can
to
take
begin.
this one. Um, so cells don't like being frozen, right? Like we want to keep the cells alive, and fundamentally we're doing something to them that they don't like. That's why I mentioned we report out post-thaw cell viabilities and cell counts, because you're fundamentally going to lose something in the cryopreservation process. Um, and there is, kind of new techniques, new media is actually the thing that we're currently evaluating. Are the dissociation, actually, so not so much the cryopreservation. We feel good about the media we're using there, but we're constantly looking at how can we most gently separate the cells from each other, because they have all these proteins, right, holding them together. Um, and in lieu of that, at least we can publish, and we have in our dissociated tumor cell user guide, what cells are what markers are known to be cleaved in our dissociation process. But that's an area that I would say is continually being examined, in order to just try to make those preparations uh, like as minimally handled as possible and as representative of the original tumor as possible. Like that's why we don't actually add any antimicrobials or antifungals. We recommend if you're culturing with these cells, you should add them. But culturing is just one of many downstream uses, and we try to meddle with the tissue as little as possible and continue to evaluate new technologies, to further that goal.